Welcome back to the biology show, and let's talk a little bit about molecular genetics and this central dogma. We've already been talking about molecular genetics already and looking at the structure of DNA. So let's take just a real quick review here. The DNA itself is a double helix, and Watson and Crick, they're the ones that we talked about that won the Nobel Prize. Now, of course, they had help. Let's not forget the help of the others, but we do want to acknowledge it. Watson and Crick are credited with finding the structure of DNA, and that they are this double helix. So looks like a ladder that's twisted. And there's a picture of it with these being the sides, alternating sugar and phosphate groups, and then the rungs of the ladder are the nitrogenous bases. All right, so we have DNA. Now you got to know the word. DNA stands for deoxyribonucleic acid. It's a great word. Remember, we talked about the sugar being the deoxyribose sugar, and the difference between ribose and deoxyribose is one less oxygen here. So it is a nucleic acid. It has two long chains of repeating subunits called a nucleotide. And we've already looked at the fact that a nucleotide is a 5-carbon sugar. It has, which of course is the deoxyribose in DNA. It has a phosphate group and this nitrogenous base. And like I said, sugar's deoxyribose. Phosphate group is phosphorus bonded to carbon, and the nitrogenous base contains nitrogen and carbon. Each full turn of the DNA helix has 10 nucleotide pairs, and the base on one side attaches to the base on the others, and this is important. The base on one side attaches to the base on the other, and so on each side of the rung, and those connect in the middle. So for example, the adenine would connect to the thymine, and it would connect there through hydrogen bonding. Okay, and our 4 types of nitrogenous bases in DNA can be thymine, cytosine, adenine, and guanine. Now the adenine and guanine make up what's called the purines, and they have a double ring of carbon. And in the model that we'll work on, it has you'll see the purines are larger, and they're a larger rung for that ladder, part of the rung for the ladder, than the pyrimidines are. So adenine and guanine are purines, and then thymine and cytosine are the pyrimidines, and they have a single ring. So adenine and guanine have a double ring. Let's take a look at those. So here's our nucleotide. Again, nucleotide is a phosphate group, a sugar, a 5-carbon sugar, a pentose sugar, and then a nitrogenous base. Adenine and guanine are the purines. They have a double ring structure, and the pyrimidines are a single ring structure. Okay, so here's your purines are your adenine and guanine. Pyrimidines are cytosine and thymine. All right, so the base pairing rules with our complementary bases. Adenine pairs with thymine, guanine, cytosine pairs with guanine here. And this helps explain how DNA replicates before it actually divides. So the question here, what is the complementary base pair for this? What I'd like to do is have you work this out, and if you bring this in to me, then I will know you know how to do the base pairing. But all you do is you match it up. Let me give you an example here. I'll do the first 3. So adenine, the base pair, complementary base pair here, adenine, you'd put a T down here. And over here with this T, the complement is A. So it would be an A, and then here is an A. And you would just continue filling out this piece of DNA. Okay, here's a picture of the structure of DNA. Again, sugar phosphates. Alternating here, 5-carbon sugar, phosphate group. It's the sugar that's bonded to the nitrogenous base. And then here we have these hydrogen bonds in between the guanine and the cytosine, or between the adenine and the thymine. And yet another picture. So however it makes it easy for you to remember, see, you can see the nitrogenous base attached to the sugar, And the phosphate group attaches the sugars together. And here's our hydrogen bonding here in the center. You are officially a DNA guru. All right, let's look at replication because we have to make copies of it. And so replication is when DNA is copied in a cell before it divides by mitosis, meiosis, and binary fission. This occurs in the S stage. We're going to look at all of that in the upcoming chapter. So just kind of plug that in your mind, know that you're familiar with it, and be ready for it. All right, so replication itself, it's going to make an exact copy of itself. We don't want to change the DNA because that changes what it is or what it does or what it looks like. And so we are going to keep it and make an exact copy of it. Now there's this process as it goes through. This process of replication is called semi-conservative. And this is the DNA unzips and one half, but the reason why it's called semi-conservative is because one half of the old strand is always kept. And this helps reduce the number of errors, during the copying process, the number of errors. If we just start, think about the game, what is it, telephone, where you start with one phrase, and by the time it gets all the way around the room, it's a completely different phrase. Well, this helps cut down on that, because if we just split it in half and just used those pieces, eventually we would certainly end up with something completely different. So by having the original strand, there's always that to go back to and make a comparison. Let me give you a picture of this. So here's the original strand here, and we have this DNA helicase that unzips this, that it opens it up. And then the new bases, these ones in green, the new bases are added. So here's an original strand, here's the original strand, and then the new bases are added. And here's the other original strand. So here's this one over to here, and this one over to here. And the new bases are then added. Again, that word that you want to remember is semi-conservative. Okay, so again, the two new DNA are formed, and they end up as an exact copy of the original. In fact, let me go back to that and let me show you. So here's our original strand, opens up. This strand and this strand are now an exact copy of that strand, that original strand. So we end up with two exact copies. All right, so DNA polymerase. Remember when we talked about ACE, we said that that was what? What was ACE? When we see ACE at the end of the word, that's right, it's an enzyme. And that enzyme, the words in front of it tells us what that enzyme does, typically. And so we have DNA polymerase. Poly means many. We're doing something with DNA, right, with this enzyme. What DNA polymerase does is it adds these complementary nucleotides that are found. They're floating freely inside the nucleus. They're inside the nucleus. Keep that in mind. This DNA replication occurs in the nucleus. DNA can't leave the nucleus. So all this occurs in it. All right, so the complementary nucleotides are floating around inside the nucleus, and they will be glued to those semi-conservative strands. So the half a rung, those pieces get glued to that through this DNA polymerase. All right, so here's a good picture. So here's our piece of DNA, the DNA helicase, helicase meaning the helix, and what DNA helicase, it's the enzyme that will open up this piece of DNA. And so that helicase comes in, unwinds it, and then we end up with this process, this piece of DNA, until there's a code that says stop this process. There's a whole lot involved with that. But here, just keep in mind this picture right here of replication. So we have an original strand, we end up with two exact copies. All right, now protein synthesis. So this concept of the central dogma, that is going from DNA to RNA, and then RNA to protein synthesis. And you're gonna know what all that means here in just a couple minutes. All right, so with protein synthesis, we have a gene that's a segment of DNA so here is that you can see this piece of DNA. Here's our chromosome, and one section of it is the gene. All right, so there's one little part. It's a segment of DNA, and it codes for some particular characteristic. It could determine hair color, eye color, height. I mean, everything of who we are is determined in our chromosomes. And so the DNA, so this DNA here makes RNA, which ultimately makes these proteins. All right, so RNA, the gene directs the making of the protein through this intermediate called the ribonucleic acid. DNA cannot leave the nucleus. First of all, it's too large, fortunately, because if DNA left the nucleus, it would be disastrous. If your template leaves the cell, you just lost all your information. And so there's a template. So RNA acts as this template. It's only single-stranded. It has ribose for the sugar instead of deoxyribose. And instead of the base thymine, it has the base called uracil. That's how you always know. If you see a U, you know RNA. If you see a T, we're talking DNA. All right, the three main types of RNA: messenger RNA, ribosomal RNA, transfer RNA. Each one has a different job. Messenger RNA is going to come from in the nucleus, and it's going to get its information from DNA. Then it's going to leave the nucleus and go out to the ribosome. Ribosomal RNA, it helps make up the ribosomes. And then the tRNA transfers the amino acids to make a protein. Now, three of these nucleotides together in the messenger RNA are called the codon. Okay, important. You'll also hear it called a triplet. All right, so here's our central dogma. Like I said, here's a good visual picture for those of you who are visual. I'm extremely visual. So we're going from DNA through this process called transcription. We're going to get to messenger RNA. So DNA to messenger RNA, double-stranded in the nucleus, transcribe messenger RNA in the nucleus. Messenger RNA leaves the nucleus and it is translated into a protein using ribosomal RNA and transfer RNA. Okay. Another great visual. Here's our DNA in the nucleus. Messenger RNA is made from the DNA, leaves the nucleus through the nuclear pore, goes out to the ribosome. Don't get confused. This looks like the pentose sugar, but it's not. This is actually a ribosome. And then you see this protein, which is a chain of amino acids. All right, we're going to look at those in more detail coming up. All right, so again, this process, transcription, is DNA making RNA. So we're going from DNA to messenger RNA, takes place in the nucleus. Translation, RNA is making the protein. Know those terms because you're going to need to know them for our lab on on Wednesday and Thursday. And every protein is made up of one or more polypeptides. Remember, poly means many. Peptide is the bond. And so we're bonding those amino acids together. Protein is a long chain of these amino acids. All right, well, error is going to occur when there is some sort of error in the DNA sequence, in the nucleotide sequence in the DNA. It's called a mutation. There's lots of ways to fix those mutations. The process is amazing, actually, to fix those mutations that occur and splice them out. But there are things that cause some escape that repair. And when we damage ourselves through sun and chemicals and things like that, then there needs to be a repair. All right. Now, these experiments here, I'm going to go very quickly through them because you have a whole nother assignment that's associated with it. But we have Griffith. Take a look at this. Know his experiment with pneumonia. And then we have Avery's experiment that came in after. And then we followed up with Hershey and Chase and viruses.